Now we'll study about another important law known as Hardy Weinberg Law, which was very important and played an important role in understanding the concept of evolution and its details. This law helped in understanding the development of population genetics. This law was given by mainly two scientists named Hardy, who belonged to England, and Weinberg, who belonged to Germany. These were the two scientists who gave the law of Hardy Weinberg that explained the concept of population genetics. In order to study the development of population genetics or this law, mainly two concepts are essential to study. First is gene pool and second gene frequency. So let us study each of them, beginning with gene pool. Gene pool can be defined as the sum total of genes that is present in a Mendelian population. Mendelian population, that is the concept of inheritance containing large population of organisms that was explained with the help of scientist Mendel. So, the sum total of genes which is present in such a population can be defined as gene pool. Moreover, a gene pool comprises diverse form of genes, that is different form of genes in a large variety. Which occurs in specific combination, forming gene combination. And recombination. By the process of sexual reproduction. Thus, through the process of sexual reproduction, the gene combination and recombination that is formed with the help of diverse genes constitute the gene pool. Then comes the second aspect or the second factor, that is gene frequency. Gene frequency can be defined as the ratio of a gene that is present within the gene pool or in a population. Of course, in a Mendelian population. So such a ratio defines the gene frequency. Gene frequency of any gene present in a population is very easy to calculate. For example, if A gene frequency contains two alleles like capital M allele and the other small m allele. These are the genes that are present within a gene frequency. So, if M allele, that is gene frequency of M allele, is represented by P and that of small m allele is represented by Q, then this calculation of such a population can be depicted by P plus Q is equal to 1. And therefore, if any one allele, that is gene frequency of any one allele is known, then the other can be easily calculated. Example, if P is known, then Q can be obtained in form of 1 minus P. Similarly, if Q is known, then P can be calculated as 1 minus Q. This forms the easy calculation of the gene frequency of any two alleles. Let us understand this with the help of an example. That is the practical application of law of Hardy Weinberg, which contains the gene frequency. In this, one has to calculate the frequency of gene within a population. So, here, as said earlier, two genes, that is M allele, where gene frequency is represented by P, and M allele, where the gene frequency is represented by Q. The frequency of M allele in the population is represented by 50% or half. That is, the two alleles possess gene frequency in equal number within a population, and therefore they can be represented as half of the total population. Condition: if the two alleles are equal in number within a population. And therefore, according to the formula, P plus Q whole square, as we know the formula that P plus Q whole square will give P square plus 2 into P into Q plus Q square. So according to the amount, P, that is half or 0.5 whole square plus 2 into 0.5 of P and 0.5 of Q plus Q square, that is 0.5 square, will give the answer 0.25 plus 0.5 plus 0.25, which represents the 25% of MM, that is 2M allele composition, is to 50% capital M and small m allele combination is to 25% of small m allele composition. In this manner, the frequency gene of the alleles which is present within a population can be calculated. The kids which we have studied within a natural population containing alleles contain the two alleles in equal number within a population. However, such a condition is always not true within a population. Therefore, when the proportion of genes within a population is unequal in number, then also the same formula would apply and help in calculating the gene frequency. For example, let us consider the gene frequency of capital N allele is 90%. That is, it constitutes the 0.9 amount. Similarly, small allele, that is very obvious, would consider 10%. That is 0.1. So on this basis, the gene frequency can be calculated as P plus Q whole square, that gives P square, plus 2PQ plus Q square, which gives 0.9 whole square plus 2 into 0.9 into 0.1 plus 0.1 whole square. That gives 0.81 plus 0.18 plus 0.01. Therefore, on this basis, the gene frequency proportion can be said as 81% to capital M allele plus 18% capital M and small m allele plus 1% to small m alleles. In this manner, the gene frequency of two alleles, either equal in number or different in number within a population, can be calculated. Now, after studying about the concept of the law, there comes significance. That is, importance and advantage of this law. According to this law, or with the help of this law, the gene frequency within a large population remains constant from generation to generation. When there is no selection or variation. Which can also be mutation. Thus, this law proves its significance within large population. But in small population, this balance or equilibrium cannot be determined or maintained. Following it, the second significance or importance is that when there occurs equilibrium population, that is maintain the population, then there is no possibility for any changes in the evolution or for any evolutionary change. As a result of which, the rate of evolution is zero. Since no evolution occurs. Evolution, according to this law, occurs only when equilibrium is disturbed. Or when the equilibrium is upset, then this law occurs or fulfills, as a result of which evolution occurs.